So let's get started. As Lionel told you, I'm going to talk to you about the how we use number and brightness to kind of trying to change paradigm in glucocorticoid receptor biology. So let me just start by a little bit about the biology. So glucocorticoids are extremely important from a pharmacological perspective. They're one of the most prescribed drugs in the world. You may have heard the name dexamethasone in 2020, since it was the very first successful treatment for COVID-19 patients, I mean, the very severe cases. And this is because glucocorticoids are extremely good on the immunosuppression and anti-inflammatory action. That's why it was so successful for COVID-19 patients. But unfortunately, chronic glucocorticoid treatment has severe adverse reactions, like the ones that you can see on this table. So one of the objectives from a pharmacological perspective is try to find what is called dissociated glucocorticoids. That means dissociated ligands that will just keep synthetic drugs that will just keep the desired therapeutic action, but it will decrease or hopefully have no adverse reactions. So in that sense, a model has been built on how these glucocorticoids work, which I'm going to be referring as a dissociated model of glucocorticoid action, okay? And this goes a little bit like this. So glucocorticoids bind to a protein called the glucocorticoid receptor, which is a ligand-regulated transcription factor. So this is a cartoon of the protein. So we have three main domains. We have the N-terminal domain, which is intrinsically disordered. We have the part of the protein that binds DNA, called the DNA binding domain. And then we have this ligand binding domain, or the part of the protein that actually binds ligand, okay? So the dissociative model says that we have two forms of the receptor. So they are in an unknown equilibrium between the monomeric and the dimeric form. And it is the dimer, the one that is binding directly to DNA. And this is historically named the transactivation activity of the receptor. So it binds to the genome and it regulates genes this way. And according to the dissociative model, it is mostly responsible for all the metabolic and the side effects of glucocorticoids. And the monomer is the good guy in the story, so it is supposed to mediate all the anti-inflammatory and immunosuppressive action, but by an indirect binding to DNA. So it forms protein-protein interactions with other transcription factors like NF-kappa B or AP1, which are major players in inflammation. So it binds indirectly to DNA by these protein-protein interactions, and it acts some sort of a co-repressor for these transcription factors. So, and that is called the trans-repression activity of the receptor, and it's supposed to do all the good actions, okay? So in a nutshell, the dissociative model states that monomeric GR is the one who triggers all the anti-inflammatory actions in a DNA-independent manner, okay? So the search for these good ligands will be to try to find these synthetic drugs that will shift the equilibrium towards the monomeric GR, which is the one that will do all the good actions, okay? Okay, and that's how this model was pursued for so long, to try to find this monomeric activity. And so how this model came to be, okay? So the key paper is from 1998, which I guess most of you haven't even been born yet. And this paper, what they do is a cell paper, and they replace the endogenous GR. They did a knock-in mice, and they put a point mutation there, okay? So this mutation right here, which is a mutation in the DNA binding domain, okay? And it's called the GRDIM mutant. This is a little more detail on the DNA binding domain of the receptor, and you can see choosing fingers, and the mutation that we're interested in is in this alanine over here, which is within this loop, which is called the distal loop, okay? So they knew from a few years ago that the knockout GR was lethal. But the big surprise was that by using this mutation that it's supposed or was known to be impairing both dimerization and DNA binding, these mice just grow up fine. And they were mostly still doing this anti-inflammatory action, and they were a little of these metabolic effects. So this huge result actually gave the rise to the name of the paper, right? Because if we have a mutant that doesn't bind DNA, and it still lives, so the ability of GR to bind DNA was not essential for survival, okay? And then by comparing this guy with the wild type, you can basically look at any pathway of GR and say whether it was was happening through the monomeric independent DNA action, or it was dependent on DNA binding and therefore the dimeric action of the receptor. But the paper that originally, and then, I'm sorry, a few years later, there was explicitly stated that if we can find ligands that selectively act via the DNA binding independent action of GR, this will be sufficient to treat basically all inflammatory diseases. So this is where the search for these monomeric dissociated ligands started to take hold. But if you look at the original data on how this GRDin protein was demonstrated to be a monomer, you go to this paper, which is the original one, and you start looking at all the figures, okay? And suddenly you cannot find any direct nor indirect evidence for or any test of dimerization. However, this was basically the base for all the search for dissociated ligands. So this is where we started working at this. So it was 2008, and Michelle and Enrico developed the DNMB assay. And so we started working on this and trying to figure out whether we can test GR oligomerization in live cells with this beautiful technique. So I'm not going to go to all the details of the technique, as you have heard it well enough early today. So I'm just going to say that so this technique uses the information on the fluctuations of intensity to estimate the oligomeric state of proteins. So if we do have different oligomeric states, like here in this example, the higher the oligomeric state that you can see, it will be seen as a change in fluctuations. Specifically, the amplitude of the fluctuations will going to be higher the higher the oligomerization state we're working on. And by measuring the variance, you can estimate how high these fluctuations were. So you can estimate the oligomerization by measuring the brightness. And so if we have you have one molecule with one GFP tag, you're going to have a certain amount of brightness, and the higher the oligomerization state, you're going to have, for example, a dimer will have twice the amount of brightness as the case before, and so on. So in our case, what we did is, so we took a series of images here, and what we did was we concentrate on a region that we are interested in, which is what happens inside the nucleus of the cell. So we keep with the pixels that only corresponds to the nucleus. So for each pixel, we have intensity levels, we can calculate the variance, and therefore we can estimate the brightness. And then we take all the pixels that corresponds to the nuclear compartment, and we basically estimate some sort of average nuclear brightness. So in that sense, what we can see is, so if we plotted the average nuclear brightness and we relative this everything to 1 be the size of the monomer, then a dimer will be close to 2 and a trimer will have a relative brightness close to 3. And so one of the important caveats of the technique is that it cannot discriminate mixtures of different oligomerization states. So if we do have a mixture, let's say we have in the same region a mixture of monomers and dimers, then we're going to receive a brightness that will correspond to the average weighted combination of those species. So in this case, it will be a value between 1 and 2, okay? So for GR, when we start measuring with these ligand-regulated transcription factors, so here are how we show the data. We have the nuclear brightness on this axis, which is relative to 1 be the GFP only, which is, we know it's monomeric, and exactly what happens with the GFP-GR in the absence of ligand. So when the receptor is not active, it is a monomer, and that's the data that proves it. And once we activate the receptor with hormone, what we get is an increase in brightness that goes from 1 to 2. So that not only means that GR it's a dimer inside the nucleus of living cells, that also means that most of GR molecules must be in its dimeric form in order to give this close to 2 value. And we can actually do a nice control experiment to test that. So let's say now we have in the same cell, I'm going to put GR but with 2 colors. So I'm going to have a GFP GR and an M cherry GR. So assuming everything is a dimer, so what we're going to have is we're going to have these homodimers of GFP, these homodimers of cherry, and these heterodimers, which if we only look at one channel, let's say the GFP channels, these guys are going to be seen as monomers because you only have one GFP per complex, okay? So here is the experiment. We have the GR with 2 colors. We only look at the green channel, and as expected, we see an intermediate value between 1 and 2 because we are artificially making everything looks a considerable amount of proteins making look like a monomer. So the hallmark or the most important prediction of the dissociative model was that GR was interacting with proteins like NF-kappa B as a monomer. So now, if we have in the same cell GR and I activate GR, and also we put NF-kappa B, in this case, one of the subunits, which is P65, and I activate NF-kappa B, then if this is true, then I should increase the population of monomers. So basically, I should get a more similar result to this one than the other to this one, because I'm basically decreasing the population of dimers. But that's not what we found. We actually found that mGR was still forming dimers even in the presence of NF-kappa B, which is most likely that we're having interactions between dimers of GR with NF-kappa B. But the whole idea that NF-kappa B was interacting with GR as a monomer came from the idea of this GRD mutation, which was supposed to form a monomeric GR, but was never tested. So when we did it, what we find was actually GRD was still a dimer, okay? And so long story short, we thought, okay, so if this mutation in the DVD was not sufficient to make a monomeric receptor, we thought, well, there must be another region of the protein that is holding the dimer together. So we mutate another portion of the protein, which is in the ligand binding domain, and that mutation enough, okay, so now we are affecting this portion of the protein, and that mutation is sufficient to start decreasing dimerization. So now we have a mixture of monomers and dimers, and when we combine, so the DNA binding domain mutation, this GRD mutation, with the one in the LBD, so we combine these two mutations, now we get what we call the GR-mon, which is a mostly monomeric protein. So we thought, well, at least these two regions are involved in making, in stabilizing the dimer. But things get from there, it got a little more complicated, because now we know from unpublished data that I was taken by my PhD student, Agustina, which is among you somewhere there. So she took another point mutation on the DVD, and that point mutation was sufficient to promote a mostly monomeric GR. So it is likely that the domains are so intertwined that the modulation on one domain is affecting completely the other. And in the same sense, we found a symmetric mutation. So now we have a point mutation in the LBD, and this mutation was sufficient again to promote a mostly monomeric receptor with just one point mutation. So that points to the idea that all domains are really involved, and they are intermingling and interacting within each other. So the take-home measure that I want to take you from here is that we see that GR is mostly dimeric inside living cells. So the idea that there is this equilibrium in the cells is likely inaccurate. And what we have is the active receptor most likely to be a dimer. So if transrepression exists, and now there are some papers claiming that this may not be the case, but if this exists, it's probably and most likely that the interaction occurs through the dimeric form of GR. So the search for monomeric GR as the holy grail of finding anti-inflammatory action is probably not going to work. And my last 5 minutes, I want to wrap up with the idea that whether we can actually say that the oligomerization state of GR on DNA is actually dimer, or is it something else? So to answer that question is not a trivial thing to do, because you have to know inside the cells where the receptor is actually bound to DNA. So we use this very special cell line. So what you can see here is GFP-GR in the absence of ligand. So you can see mostly in the cytoplasms and a little bit in the nucleus. So once we activate the receptor translocates into the nucleus. And I hope you can see my mouse, but this big block over here, which is something special in the cell line. And oops. Sorry, my computer just hung out for a second. Here we go. So we got this big blob, and what we have there is we have a gene cassette. Okay, so we have this sequence, 9 kb sequence, where they have binding sites for the glucocorticoid receptor. So, and this is repeated 200 times across in the same region of the genome. So basically, we have a big array, a big attractor of highly specific binding sites for the receptor. So we get a high concentration of molecules. So we are basically able to see where GR is bound in live cells. Okay. So the number and brightness has spatial resolution. So we can ask, what is the oligomerization state of the receptor in that concentrated region when we know it's binding to DNA? So here I'm showing you the results in the nucleus. So in red, I'm showing you the entire nucleus. You can see that it's a dimer with the natural ligand corticosteroid and even with the synthetic rock dexamethasone. But the question is, what happens at that specific point where we have a lot of DNA binding? And we were extremely surprised to find that the number is higher than 2. It's a number between 3 and 4. And it goes straight to 4 when we added the synthetic drug. So the most parsimonious explanation of that is that GR is predominantly a tetramer, at least at this structure. So we spent actually quite a few years trying to prove otherwise. So whether we can, this was some kind of artifact. And so I'm just going to show you one experiment that we did that personally convinced me that we actually had something rather than an artifact here. So these guys were studying the DNA binding domain only part of the protein. And what we have here is what it looks like when it's bound to DNA here by X-ray crystallography. And here is what it looks like when it's not bound to DNA, measured by NMR in solution. Okay, so you can see that the structure is quite similar. The only difference is right here in this part, specifically this, which is the distal loop, which changes conformation upon DNA binding. So I don't know if you can see my hands. So basically what I'm saying is that so the receptor has one conformation, and once DNA binds, the receptor changes conformation, okay? So it just shifts. So these guys find a point mutation. What they do is they stack the receptor into the DNA bind conformation, even though it's not bound to DNA, okay? And this was fantastic for testing our hypothesis that it's somehow the receptor, once it's bound to DNA, was getting some conformational changes, and that was triggering the tetramerization of the protein. So we got this mutation now that it's triggering the receptor into believing it's bound to DNA when it's not. So we put it in cells, and at the array, we get the similar 4-ish values. But the question was, what's going on in the nucleus, in the entire nucleus? Because what we are basically measuring in the entire nucleus is the diffusive one. And now we're just triggering the receptor in the DNA bound conformation. And we were extremely surprised and happy to find that that point mutation, so making the receptor believe in it or having the DNA bound conformation, was sufficient to make a tetramer everywhere in the cell, okay? So we did a lot of mutations and truncations, and I'm not going to go into the details. I'm just going to show you that what we came up with is that we have a model where GR it's a dimer with at least 2 dimerization interfaces. Even though this intrinsically disordered region is also involved somehow in dimerization, it binds to DNA. Once it binds to DNA, there's some conformational changes supported by X-ray crystallography, and that somehow triggers a conformational change in the LVD, so in another portion of the cell. And the LVD is necessary to promote this tetramerization. And the tetramerization, we don't know exactly the shape of it. We don't know if it's head to tail or a head to tail configuration. And it's very tempting to speculate that if the head to tail configuration is the one that is happening, that will likely promote the looping between 2 different parts of DNA, which we know it's important for gene regulation. So in summary, what we are claiming is that monomeric GR is not physiologically relevant, at least in our hands, that dimerization depends on all domains of GR, which are extremely intertwined and they connect to each other. And we propose that the active GR, so the DNA bound version, is likely a tetramer. And we believe for all these reasons that the dissociative model should not be further pursued. So with that, just another model, we have the dimeric form of GR, which is the active form. And once it binds to DNA, it goes through a complex cycle of forming tetramers and then dissociated into dimers again. With that, I just want to thank a lot of people. So